I'm really excited to present some of our work here and to have like interesting discussions with all of you. So I hope topic-wise it's a fit. It's probably quite broad, but I'm kind of hopeful that like we'll have some interesting discussions later on. And so I'd just like to super briefly start with like how my lab works. We're very focused on clinical needs. So all our projects are motivated by clinical needs. And this is basically the starting point. So we are quite technology and field agnostic because we basically work towards solutions that fulfill these clinical needs. And our core expertise is in materials chemistry and engineering. And so we try to really develop solutions that are tailored to these clinical needs together with clinical partners. And so our aim is to go from the initial idea concept to the late preclinical stage where the technologies are then basically taken over by either a company or a spin-off that we form. And so a few years ago, I had a lot of interactions with gastrointestinal surgeons. My best friend is a gastrointestinal surgeon. And so this led to awareness of problems that gastrointestinal surgeons face in their daily life. And so I think many of you know that unfortunately about 20% of us have to undergo gastrointestinal surgery at some point in our lives. And this can be due to inflammatory bowel disease or tumor removal or other kind of like pathologies. And so what the surgeons do is they resect the diseased segment of the bowel and they reconnect the healthy tissue with a suture line, and that's called anastomosis. So this usually goes well, but in around 10% of the patients, this suture line or the wound doesn't heal and a leak develops. And that's obviously quite serious because you have gastrointestinal content that contains digestive enzymes and bacteria, and this content leaks into your abdominal cavity and starts digesting your own organs, essentially. And so this is a super serious complication. It's very deadly and results also in high costs for hospital. And so a few years ago, we thought that we can develop a solution to prevent this complication from happening. And so we developed first a polymeric adhesive that can seal the anastomosis and reinforce the anastomosis. And because we can achieve very strong tissue adhesion, we can also place some sensors there that can indicate healing complications. And so just a little bit of chemistry, we call this new adhesion technology a platform technology because we can basically anchor a biomaterial, a hydrogel, by forming a polymeric network directly on the tissue by photopolymerization. And in this way, we can anchor a hydrogel directly on the tissue really well. And so the adhesion is much, much stronger than what you typically get for mucoadhesion or for any kind of interfacial cross-linking. And so because we can keep these adhesives there, we can also include sensors into these adhesive polymers. And the sensors in this case are chemical sensors. So they're electronic free and they are super simple. Basically, we can put the carbonate, in this case it's barium carbonate, into the hydrogel. And if the barium carbonate is present as a salt, it basically doesn't show a lot of signal in the ultrasound. But if the pH decreases and the carbonate is dissolved into CO2, these bubbles get entrapped in the hydrogel, and this becomes super visible in the ultrasound. We can also put proteinaceous gas vesicles. These are gas vesicles that are produced, for example, by Mikhail Shapiro's lab at Caltech. And these vesicles contain gas and they contain a proteinaceous shell. And if there are enzymes present, like digestive enzymes, proteases, for example, they can cleave these vesicles. And this basically leads to the loss of the gas and the loss of the ultrasound signal. And so we can also implement that in vivo. So we perform pig studies, preclinical studies, where we attach these sensor-containing hydrogels on the pig intestines and stomach. We can observe suture breaches very easily by very affordable point-of-care ultrasound devices, such as the one shown on the left. So it's basically an ultrasound scanner that you can buy for $1,000 and connect to your smartphone. And so you can monitor the suture site. So this is an intestine, a cross-section. You can see the hydrogel patch, and when the suture is intact, the patch stays dark. When there's a suture breach and intestinal content comes in contact with the sensor, we can see the sensor light up. And to make this more easily visible, in the abdominal cavity, we can also change that into shape morphing elements that are visible in either ultrasound or CT. So these are basically the two clinically used imaging methods to assess anastomosis, the intactness of the anastomosis. And so when the suture line is intact, these elements basically stay the same. And when there's gastrointestinal content in contact with the sensor, we can see that, for example, there's a ring that disappears and gives contrast in the ultrasound, or there's a line that disappears and the contrast disappears from the computer tomography data. So of course, this would be nice if we could bring that into clinics, but it's a medical device class 3, so it takes a bit of time to bring that into patients. And since this gastrointestinal leak problem is massive, we thought that we can provide the hospitals, patients, and doctors with an intermediate step. And we can also implement these sensors into drain bags that are typically attached to the patients after surgery, and we can use the exact same sensing elements that are cleavable by digestive enzymes. We can attach these sensing elements onto drain bags and then monitor the content and the presence of digestive enzymes in these drain fluids. It's not perfect, because once they end up in these drain fluids, it means they're already present in a patient's abdomen, which is not ideal, but it's still better than the current clinical gold standard. And so with this technology, we also validated that in patients. So we put these sensors, which are essentially gelatin, starch, and lipid based, and colored with some pigments. We put them into drain bags and brought them in contact with patient drain fluids. In this case, it was a small study. We had 6 patients with a leak and 26 patients without. And for the trypsin responsive element and the amylase responsive element, we got perfect diagnostic performance. So we detected all the leaks and no false positive and no false negative. So we were super happy with the outcome of this technology, of this study. And now more hospitals are joining. So hopefully this will allow us to gather the data for then further clinical translation. But this led us to ask the question, if we can not only address the problem once it's developed, maybe we can also make surgery better and prevent these leaks from even happening, right? And I mean, it's maybe not the entire story, right? These leaks don't only happen because of bad surgery. There are also many other factors like patient comorbidities that lead to these leaks. But it's still kind of bothering that if we want to reconnect these ultrasoft tissues of the gastrointestinal tract, that we pierce them with either staples or needles. And we thought that maybe we can reconnect tissues a little bit more elegantly and gently. And so instead of what we're doing today, we thought maybe we can do something similar to metal soldering. But of course, not using a lead solder, but using a protein-based solder. And we can maybe solder soft tissues together by heating a protein-based solder on top of the tissue and achieve suture-free reconnection. This is particularly interesting for fragile tissues. Just like in electronics, we also solder together fragile electronic parts with this technology without damaging them. It's very similar for tissue. And it's especially interesting also for robotic procedures, because typically for the robots, it's super hard to understand like the tissue properties, right? Because there's typically a little haptic feedback. And so it's very, very hard to suture soft tissue well. And so we developed a solder paste that contains albumin and gelatin. So it's basically the proteinaceous and form-giving backbone. And we added nanoparticles. It can be gold or it can be titanium nitride that allow us to heat the paste very efficiently with a laser, for example. And we included nanothermometers. These are nanoparticles in this case that emit fluorescent light depending on the temperature. And so we can measure the temperature based on fluorescent emission in a contact-free, non-invasive way. And so with this paste, and I think I'm going to show a few surgical images. So if you're sensitive, maybe that's the time to quickly close your eyes. We can place this paste in a setting that's either open surgery. This is a pig. And we can heat the solder paste with a laser and fuse together the tissue. Or we can also implement that into either laparoscopic or robotic surgery. It shows that the solder is really beneficial, especially when it comes to ultra soft tissues such as intestine or the liver, where sutures underperform, not because they break, but because they basically cut through tissue, like the string that you maybe need for to cut soft cheese. It's basically what happens with the suture and soft tissue. And so this can be also implemented in robotic procedures. Very, very easily, the robots can solder and reconnect tissues autonomously in a feedback-controlled way. And in this way, really achieve gentle tissue reconnection. And so I hope in this first part, I could very briefly show that there are a lot of surgical problems that are still unresolved, and that materials innovation can really offer some potential solutions, hopefully. We see that especially new tissue adhesion technologies that perform well under wet conditions, under very difficult conditions, are especially promising, because there are lots of patients that have non-healing wounds, and there are very, very limited options available for these patients. And so in the second part of my presentation, that's hopefully a bit shorter, I'm going to very briefly discuss two different technologies in another field, and it's a field of women's health. These projects were very strongly motivated by a gynecologist that joined our lab as a research fellow, and he really brought a lot of clinical expertise and clinical needs into our group, and he worked with physicists and chemists and engineers in our group to develop some solutions for these women's health clinical needs. And so I think this one is for me was super shocking, because I didn't know that female contraception was still basically solved by pretty archaic approaches. So we either have hormonal contraception with its downsides, or we have basically tubal ligation, which is a pretty invasive, usually non-reversible kind of intervention. And so the gynecologist wanted us to develop a reversible contraception approach based on a hydrogel. And that's, is, from a materials point of view, is probably fairly doable. But what happened in the course of this project is that we discovered that such a tubal occlusion with an implant is not only interesting for mechanical reversible contraception, but potentially can also prevent the backflow of menstruation blood into the abdominal cavity. And this is a process that's called retrograde menstruation. And it's believed that it plays a key role in the development of endometriosis lesions. So cells, endometrial cells flow back with the blood in the abdominal cavity. And they don't grow in all patients, but in some endometriosis patients that have a genetic predisposition, some of these cells grow in and then form these endometriosis lesions in the pelvic cavity or the abdominal cavity. And that's still a hypothesis, but it's a prevailing hypothesis. And so we thought that we can basically develop an implant that solves, or at least attempts to solve, both of these problems. And so we developed or designed the implant in a way that it can also be removed by either light or a fluid. And so we defined some design requirements. So we wanted the implant to prevent transport of cells or sperms. We wanted the implant to be reversibly removable. We wanted to be able to place the implant in a non-invasive way, and of course, biocompatibility is also super important. And so we chose an acrylic gel that has very, very high swelling properties. And I show that in a minute why. So initially, the gel has the form and the size of a rice grain. And this can be placed hysteroscopically. So this is basically the endoscope of a gynecologist. Can be placed hysteroscopically into the fallopian tube. And then it basically just absorbs fluid, orbital fluid from the surrounding. And it swells a lot. So the scale bar is different, but it swells like 5 to 10 times and occludes the tube. And so when the contraception is no longer wanted, we can apply a stimulus such as light or a glutathione fusion solution. We can fully liquefy the implant and basically dissolve it. And so, of course, in the translation, it's super important that this implant is compatible. And so we performed extensive cell compatibility tests that look good. We also performed a pig study that showed that we can keep the implant in the tube, and we don't really see any kind of adverse reaction to the implant for a limited time. It's 3 weeks now. It's very expensive, so we had to stop the experiment at some point, but it looks fairly promising. And so most importantly, we can also occlude the transport of cells and sperm. And so if we put the implant, the tube is really fully occluded. And the last project that I'm going to show super quickly is also motivated by this gynecologist. We also found that basically the entire area of women's health is very under-researched. And as part of it, also menstruation blood analysis is not fully exploited. And we thought that we can change that, and we can use that as a valuable biofluid that can allow us to monitor health and disease in the menstruating population. And so we developed a super simple assay, but it's basically no rocket science. We implemented a lateral flow assay and a volume control, and this is the main innovation. So we developed a microfluidic chip that allows us to control the volume that ends up at the center super well. We can embed this into a hygiene pad and then really do in-pad analysis of biomarkers. So in this case, we have a proof of principle for CRP for 2 cancer-associated markers and CA125, which maybe also can indicate endometriosis in some patients. And so really the key is to control the amount of fluid that ends up at the sensor, so we can also semi-quantitatively analyze the biomarkers. And then in order to assist the users, I think we've seen in the COVID test that sometimes can be super confusing, and then if they have a line, they don't remember if it's negative or positive now negative, or, you know, we developed an app that also guides the users through the process and the interpretation of the results. And so the idea with this technology is that it's really a screening tool. It's not gonna give a diagnosis, but it's gonna alert the users to maybe get a further checkup. And so with this, I have the second part showed a little bit that there's a lot of room for technology innovation in the women's health domain. Of course, I would like to thank all the people in my team that actually did the research, all our collaborators, also many of them clinical collaborators and funding agencies, and I'm very happy to